All right, so today we're going to be talking about like the structure of skeletal muscle. So first, it's important to know that our sarcomere is the functional unit of the skeletal muscle. So that is just what's between two C lines. So like right here, this would be a sarcomere. So yeah, we can see kind of that. And then our A band and our I band. So our A band is this dark band here, and our I band is that light band. So that's what gives rise to the striation patterns. All right, moving on down here. So we are able to see that like more in depth of our bands. So this section right here is dark because of overlap. And that's overlap between the thin and thick filaments. So our thin filament is actin. And I like to think actin kind of makes me remember actin is the thin filament. And then the thick filament is myosin. And yeah, and then so here we see our M line as well. This is thick due to additional proteins. And those additional proteins are just what kind of link the filaments together. And so that's why it just kind of makes it thick all right in there. Yeah. So then if we move on down here, we see the pattern of our sarcomere, like a cross section of it. So, right, cross section. And we see that there's a difference in our thick and our thin filaments. So our thick filaments are going to have, or for one thick filament, there's six thin, and for one thin filament, there's three thick. So our thin filaments are double our thick, basically. So I'll write that here. Thin filaments, double thick. So you'll have for one thick, you're gonna have two thin. So perfect. And these numbers just come from down here, like this diagram. You can see how there's one thick and six thin, or one thin and three thick. So if we move down here, we can see kind of our movement, it's called the sliding filament model or mechanism. And that's just where we can see that over here, they're lengthened. There's a lot of room in those corners. And down here, they're completely shortened and they're all really tight. Now, it's important to note that these filament lengths have not changed. They just slide over top of each other. So that's an important concept to remember. And then the main picture down here. So this is super important. Like how does that slide? So down here we have our actin, our thin filament, and our thick myosin filament. So our thick myosin filament has the globular heads that allow it to cross bridge, which we'll get into in a different video. But here we're going to focus kind of on our tropomyosin and our troponin. So our tropomyosin is basically what covers the binding sites. So I'll write that. Covers binding sites. And so this prevents contractions. And our troponin is actually what has a calcium binding site. And so once calcium binds, then um, our tropomyosin will move out of the way so that our globular heads can bind. So, all right, move or make tropomyosin move. And yeah, and so we'll get into cross pressure cycling in another video.